This is probably one of the harder talks I had to do. A, what have you learned in 5 years? Well, quite a few things, but how do you capture them such that you can convey that in 5 minutes? Because I was told in no uncertain terms by some people in the audience that I would be cut off after 5 minutes. So I just decided to condense things a bit more. Well, start with the rationale, which I guess with all of you is something you're very familiar with anyway. But just to remind you that what the rationale is, that is the idea that we hope that using nanocarriers would allow us to increase transport of peptides across the epithelial barriers in the gut. And why we bother with nanocarriers is, of course, that there are penetration barriers, there are barriers in terms of stability, and we hope that essentially what we're trying to do will work, which is to replace the interaction which we cannot control, which is causing the problem and bringing about the sort of limitations, the interaction between the body and the drug or peptide with that of the carrier and body. And I guess ultimately to release the drug and in that way have dealt with delivering the peptide. So that's the principally the idea, and that idea also informs, of course, then how you set up the project. So you hope to have certain functions that the nanocarriers should provide. So one is, for example, encapsulation. Obvious, but not actually trivial, as it turns out. Often for hydrophobic drugs, you would talk about solubilization. We want to be able to localize the carrier within the part of the encapsulation idea is, of course, around the protection. And this is a sort of, with hindsight, somewhat naive slide I've been using for many years. And one of the things I've learned that in these 5 years that it is a naive slide, because there's a big part missing here, which I think would be quite important to understand in more detail, and that's where I end up with this talk, actually. So protection, and we're thinking about the enzymes and pH changes here. Then the idea is to penetrate the mucus, as we've heard a lot about this morning from Justin, and then I guess we think we need to get into the vicinity of the epithelial cells. And one of the earlier points of discussions in this consortium was around the idea, well, what happens then? Are we happy if nanoparticles are going to be taken up, or do we hope that the peptide is going to be released there? And will we have interaction particles going through there? So these were questions that we, at the outset, thought would be important, and that informed, in a way, how we used our building blocks. So the building blocks, as Noemi has already sort of explained, were around the various prototypes, as we say. That is the basic systems for making nanoparticles, and we had initially divided them into nanocapsules and then others, i.e. complexes, particles, and micelles. And so we had a good selection, actually, of different carriers. It turns out, though, that there were some limitations that sort of come about, we learned about, understood and appreciated, that if your peptide has certain physical chemical properties, obviously, then that leads you to make choices about how you can encapsulate. And I guess that over the course of the project, we took on more peptides, but also more carriers to be able to combine those in a more suitable way in many cases. So we have these different building blocks. One of them, of course, is the peptides. And here, I guess this was very much something informed by our large pharma partner, Sanofi. And I guess their considerations were around learning, but also potentially, I guess, in terms of markets and how something like that could be used later. That meant initially we focused on a sort of limited range of peptides. And we did, however, have peptides that had very key differences in some key physical chemical properties. One, what that we turned out to then have a broader range actually was around the isoelectric point, the size of the molecular weight of the peptide, And then here, quite important in terms of matching up with the prototypes was this, well, are they hydrophilic, are they hydrophobic? Because, of course, nanocarriers very often will use some hydrophobic interactions to form the nanocarriers. And one of the challenges that we dealt with was that a lot of the peptides were very hydrophilic. And so some of the learning that had to go into this was actually the adaptation of this matching physical-chemical properties of peptides with what the different types of nanocarriers could do, and then throughout the course of the projects to bring in new prototypes that could accommodate these peptides. And I think these were key steps and decisions that we took that allowed us in the end actually to deliver some interesting in vivo results. So we had a broad range of molecular weights, and one of the key issues that sort of are key decisions in this whole process, you saw the extreme funneling that we had to do because, of course, as you move towards in vivo and then potentially further, you have to limit the choices of systems that you work with just because of the resource limitations. So our understanding that we discussed before about how we think uptake will work also then informs the types of assays that we use and develop. For me, another key thing that comes into it is, and that is I think where some of the key learning came from, was that people with very different experience and background in terms of drug development and development of nanosystems, i.e. academics and industry, met up. And we had to establish a dialogue and understand the concerns that come from the different areas. And that's a key thing, I think, learning that happened. And you can see it's not just the physical chemistry and how big should they be, and should they be positively charged, negatively charged, or mucus penetration. A lot of other considerations have to be taken into account when we decide how to funnel these things, how much loading do we need. Based on the disease model, for example, what is our expectation of how much dose we can give? How much commercially do we need to expect of these peptides to be actually delivered in order for it to be a feasible project potentially? So it was in this interplay between long-term vision about how a product could look and having input from pharma and the academic research questions, I think, where a lot of the fruitful interactions came around this project. And that ultimately shaped and changed the way we looked at how to funnel and assess the different, you will not learn what I learned. Anyway, so this learning went into the target product profiles. So essentially decisions around what we think these nanoparticles should be looking like. I'll keep talking anyway. So one of the key things is that they provide guidance. And Justin asked that question about how do you decide which one should go forward. I think a key insight was that we didn't exactly know which models would move forward. And therefore, we wanted to have a library of assays that would allow us to understand how systems behaved, to also be able to go back and decide, well, which assays actually are meaningful. So having this as guidance rather than a firm set of rules, I think was an important decision. Others were already talked about, the SOPs. Here, there was a learning, a compromise between how standardized you want to do something, how standardized you can do something, because for some assays, for example, encapsulation, the individual requirements of the carriers will be very different, and you cannot have a standard. But you can say, well, what does the assay have to deliver us in terms of information? And we certainly tried to standardize that if we couldn't standardize the assay itself. So we've talked about this, so I'll skip most of that, but key was that we learned how to match up peptides and prototypes. New ways of formulating, new prototypes were introduced. We learned that in this large network we had to work with standardized prototypes. We did that, and protocols. That worked out well. And ultimately we produced prototypes and information that will allow us to make the link back which assays are actually delivering meaningful information. So in terms of other things, well, this interaction between with the academia and industry, I think, was a key thing because there's key learning there in terms of also the scale up. So it's a lot of information exchange there. What are the requirements? How do we define the process so that they are robust? This is learning that has gone both ways, I suppose. And we were able to develop in expectation of thinking about what ultimately the dosage form should be like, gastroprotective dosage forms for all of those candidates we wanted to take forward, so that they could be potentially given in vivo. And then ultimately, I guess we'll hear about more about this, is we found that we have prototypes that work. And this is what my key learning is really this, where I say this was naive. So that ultimately something that's very difficult to capture and where we do not know what is important and where we have to go through what we've learned now and sort of reassess is the whole interaction of nanoparticles in the GI tract. The mixing, we have different pHs, we look at different species, they have different pH gradients. All of those components, I mean, if you look at the array of different stimulated gastric fluids or stimulated intestinal fluids, they all differ in some components. They may seem trivial, but if you have cholate in there or not, it will make a difference. And that's something, I think, where we need to understand a lot more about how to simulate that. I apologize for running over, and happy to take any questions.